Hello, everyone. I'm Jared Fagan. Sorry I couldn't be with you in person today, but I'm going to be talking about the sequencing cost and the bioinformatics processes involved with Oxford Nanopore sequencing. So the cost of Oxford Nanopore is relatively low cost. You're looking at about $500 for a quality flow cell. Their best deal is the $1,000 starter pack, which is for the mini ion device, which is their smallest sequencing device. So it comes with the device itself, two flow cells, the library preparation kits, which run approximately $50 to $100 per kit, and then access to the community forums. So you can get questions answered by professionals from Oxford Nanopore, raise concerns, talk with other people who use the technology, and bounce ideas off of each other. The company actually ran 5 flow cells at once at 11x coverage, and coverage is defined as the number of sample nucleotide base sequences aligned with specific locuses in a reference genome. And this is calculated by taking the read length and base pairs, multiplying it by the total number of reads produced by the sequencing, and then dividing that by the haploid gene length. So at 11x coverage, 5 flow cells ran for $2,750. Now at $500 per cell plus $50 to $100 per kit, it stayed pretty on cost. And there's two main reasons that the cost is relatively low for this. The first being that it's a small compact device, especially with the mini ion. Now the company does sell some larger instruments to sequence with, including the grid ion, which costs around $50,000, and the Prometh ion, which costs $10,000. These products are much bulkier and can analyze multiple flow cells at once, where the mini ion, you're just looking at one flow cell, and they can generate up to 250 gigabytes of data. But the regular mini ion, which most people seem to be sequencing with, unless you're an industrial type of sequencer, is relatively low because of how small and compact it is, and it also doesn't need a dedicated sequencing instrument. So this device basically runs on its own. You don't need any other instruments. You don't even really need a computer to process and analyze all the data. It's going to do it itself. And then in terms of this bioinformatic processes that Oxford Nanopore reads can be used for, there is a large list of them. Some examples include quality control, which examines the distribution of quality scores using a computer algorithm along the sequence. DNA and RNA modification detection can be used, base calling, which was referenced earlier, as well as transcriptome analysis. The figure showed on the slide here looks at all the different analyses taken from a mini ion sequencing technique and how a group of scientists broke those down. So you see de novo genome assembly, error corrections, base calling, quality control. All of those were performed by the mini ion and the group of scientists based off one sequencing read. Oxford Nanopore can also perform and investigate epigenetics and methylation analysis, along with chromatin conformation, fusion transcripts, single nucleotide variants and phasing, gene expression, and structural and splice variation. The sequencing can include the whole genome. They can also target certain sequences, do the whole transcriptome. There's a short fragment mode included, and then they can also look at metagenomics techniques and microbiome analysis. As for genome assembly, full genome assembly is definitely a strong point of Oxford Nanopore, especially when there is third generation sequencing reads used. These third generation sequencing reads improve the quality immensely compared to the second generation sequencing reads. And this is because when we look at the repeated regions with third generation sequencing reads, they're often expanded, and that thus lowers the error rate. And when we look at error rates, a lot of the error rates for all sequencing technologies occurs due to repeated regions within the DNA you're looking at. Second generation sequencing reads will still produce a result, but the error rates may be high, and it may not be what you want. For ChIP-seq and RNA-seq, in my research, I found that Oxford Nanopore actually can perform these directly using the instrument, and they state that this allows for accurate quantification and complete full-length characterization of native RNA or cDNA without fragmentation or amplification. And what this allows for is streamlining analysis, and it removes the potential for biases within the data. And here are the works cited.